I just did.
There we go. All right, sweet. Well, I appreciate you guys joining on a March Madness Sunday afternoon. Wasn't thinking about the date when I booked it, but Ohio State is probably about finished getting their tush whipped by Villanova right now. So I'm glad I'm doing something different anyway. So it's really a pleasure to be here. And I've prepared some slides for you all, but since there's so few of us, if you want to interrupt or say something in the chat or make it, just nice and casual, that'd be awesome. I'm just going to share my screen here, since we've all figured out how to do this during the last 2 years. So what I thought I'd like to talk about today, since we've had a tough 2 years of a pandemic, is to try to give people a little bit of hope that there's not just all nastiness in the world, and talk about the good side of viruses. And as Ray said, I'm at Ohio State University now, or was from Maryland, the free state of Maryland. And now I'm at Nationwide Children's, affiliated with the Ohio State University. If you want to follow me on Twitter, it's at KidsOncDoc, or feel free to drop me a line on email, timothy.cripe@nationwidechildrens.org. I'm happy to connect with people. So, I think all people on here know that, we recently lost a giant in NYSC history, Stormin' Norman. So I thought we'd just take a moment to remember him. And if you want to put any memories in the chat or chit chat or afterwards, just last week, sorry that we've lost him. He was an inspiration on the Seneca Rocks, and many other places have fond memories of all he taught us. So I, like any good science camp event, thought we would have a little fun, in this case, fun with nostalgia. We'd also touch on a little science and a little bit of philosophy. So part one is a little history and nostalgia. So I wanted to thank Ray for inviting me. Here's me serving Ray and Rod at the Lord Dill Dining Supper Club. And, I'm trying to be as sophisticated and suave as I can be. So I have Pete Samard to thank for all these photos I'm going to show you, because I was able to dig out a whole bunch of nice 5x7 black and white prints that he stashed away with me years ago, and I was able to still find after all these years. So I thought, since I'm giving a lecture today, I would show you the first photo the only photo I have of myself giving a lecture at science camp. I remember doing it outdoors, but looks like I was more dancing than talking. So hopefully that was entertaining. But I spent most of my time playing Frisbee, as you all know, and that was always a lot of fun, setting up Frisbee golf, playing on the green. So this is my photo out of our yearbook that year. And, 44 years does a lot to a person, a lot of changes, but a lot of things have stayed the same, I would say. I was eager, optimistic, happy, and clueless at the time. And when I first made this slide, I thought, well, I'm still eager, optimistic, and happy, but I feel like I'm not clueless anymore. But then again, life, I was always hitting you with something. So today I took that out and said, well, I've got some clue, but there's still a lot of things I'm clueless about. It's a tough world for sure. So I just thought I'd review for you, by way of introduction, sort of where I went after science camp. And because, I was always wearing the t-shirts. I was at Princeton for undergrad, spent 4 years there. I went out to the University of Iowa to become a Hawkeye, to do my MD and PhD in genetics. That took 7 years. I stayed there for 3 years to do a pediatric residency, met my wife there, Linda. Then I moved to Boston and did a year at Children's Hospital in Boston for a clinical year. And then the person I decided I wanted to work in the lab to be my mentor, he decided to move to the University of Colorado. So I followed him out to Denver. So that was my career gestation and training, 17 years after high school. Then my postnatal career development, I got my first job at the University of Wisconsin in Madison. became a Badger, was there for 4.5 years. Then I got recruited to the University of Cincinnati and Cincinnati Children's Hospital, was there for 12 years, kind of climbed the ranks of academic medicine, enjoyed my time there. And after 12 years, got recruited 10 years ago to Nationwide Children's Hospital up here in Columbus. So I've been in my postnatal career development for 27 years after 17 years of training, and still going strong and enjoying it. So let's move on to the science a little bit. What is a virus? Just a couple of slides about that to get us all on the same page. So Nobel Prize winner Peter Medawar said viruses are a piece of bad news wrapped in protein. And that kind of sums it up, I think, for most people when they think of viruses, what they think of. And viruses come in all shapes and sizes. When I was in grad school at Iowa, my favorite course was virology, just because it was so fascinating. I came aware from that course thinking, if you can imagine a way that something can do something, there's a virus out there that does it that way. All these viruses are different sizes, different shapes, different compositions, different proteins. Some are DNA, some are RNA. Some have lots of proteins on their surface. Some are covered with a membrane. They're just all over the map, all kinds of shapes and sizes. What I'm going to talk about today are some of the smallest ones. Parvovirus down here. Adeno-associated virus is a parvovirus. A little bit of adenovirus here and some herpes virus here. All sounds like fun, I know. But we're gonna get into why those could be not so bad. Maybe some good. But we know there's, like I say, tons of viruses. COVID's not the only one. They're all over the world in endemics and causing lots of havoc. We see them every year in the hospital with the kids getting all kinds of different viruses, wreaking all kinds of havoc. I don't need to, I know you all know they're bad. So hopefully I can convince you that there's also some good. And that's because of these properties here. So there are really two fundamental types of viruses in terms of the effects they have on the cell. And I categorize them as lytic or non-lytic. And here's some examples of non-lytic viruses. They're viruses that mainly just put their genes into the nucleus or sometimes into the cytoplasm, and then they can produce more themselves, but they don't kill the cell. They turn it into a virus factory, or they go latent, like herpes virus, and they're just in there ready to do something at some point, but they don't kill the cell. And that's really important because we can leverage that and use that to our advantage. The other category is the lytic ones. These kill the cell and they make more of themselves. So they're more destructive. And I've listed some of those. Adenovirus causes common cold, herpes simplex, as you know, the cold sores, measles, polio, SARS-CoV-2 is lytic, lots of others. And this is also an important property that we can leverage and use to our advantage. So I know it doesn't sound like something we should be messing with, but we can do it. So let's take some viruses, we'll make some music, we'll do some magic like we did at science camp. Woody and Dave Villanova, and speaking of Dave Van Pelt, harmonizing with our nicely racially appropriate colored mustaches, I guess, and ties. Classic photo, lots of fun times. Or Bob leading us, you know, the mini tiger tones on Rob, Harvey, Raju, Todd, leading us on the street walk, singing My Girl. So some good times there. And making magic, of course, Joe, Shoemaker, Raju, doing some magic at the supper club. So let's see, how do we use viruses to make some music and do some magic? So let's talk about those non-lytic viruses and how we've used them for medical therapy. So I'm going to tell you about a couple different diseases. So there's a disease called Leber's congenital amaurosis, which is a congenital blindness that you have missing this protein RPE65 in the retina or have it mutated. And this is a measure of your sight. And you can see this dotted line is, or the line up top here is 20/20. And then 2200 is legal blindness. 20,000s unable to perceive hand motion. You can see that as these kids age, they start losing their sight. They're legally blind in their young teens, and then pretty much can't see a thing in their later years. But this is really a disease that's been markedly changed by the use of viruses. So there's a technique that was developed called multi-luminescence mobility test. So this is where they actually build a room, have the kids or the young adults walk in to the room and have to step over steps, make turns, stop at a stop sign, step over more steps, and so forth. And they can adjust the level in the room of lighting. And you can see here, from 400 lux, nice and bright, shown over here on the right, down to a 1 lux, a moonless summer night right here, that if you have 20/20 vision, you'll still be able to navigate. And so what investigators did is they took that little parvovirus, that tiny virus I told you about, adeno-associated virus, and they put in the gene for that that protein that was mutated, and then injected into the retina of the eye. And you can see that this is at the time of or before they get treated. These are different various time points across the bottom before they get treated, and then they get treated, and then time points later. And you can see how bad their vision is down here at 50 lux, where they can't navigate that at any higher. Actually, so half the group got treated here and half the group got treated here. So the group that got treated at the beginning in the blue, right away they were able to navigate that at much dimmer light. And there was a control group that they kept following, and then they treated them later. And as soon as they treated them, you could see you could see that they can see. That's pretty amazing. Pretty amazing.
Car, get my clothes. What I'm looking for might be in there.
So that became the first FDA approval of a gene therapy in a person in 2017. They call it Luxturna. You can, Lux is the light, Turna, RNA, produces a protein. It's got a complicated name here. They give it subretinal, and the blind can see. They only could do that because they used a virus that is able to get a gene into a cell and have it correct that cell. Spinal muscular atrophy. It's a neurologic disease where you have a mutated or missing protein called SMN1, spinal muscular neuron 1, SMN1. And normally the nerves innervate muscle. When you don't have this protein, the nerves degenerate. They don't innervate muscle. The muscle gets weak, and that includes all your limbs, your diaphragm, everything else. So at birth, this is all pretty much intact, although not totally, but it's starting to degenerate, but it's there enough to get you to birth. And then you can start seeing you gain some milestones, head control, and so forth. But then in the type 1, the most severe type of disease, you start losing your ability to control your head or sit. You can't walk, you can't breathe. And without a ventilator, you die. And type 2, there's more mild types that take a little bit longer for you to deteriorate. So I'm acutely aware of this because this treatment was invented at our hospital here in Columbus. Again, taking that small virus, adeno-associated virus, and delivering that SMN1 gene. And in this case, they actually give it into the vein, a single infusion. The virus goes to lots of places in the body, but it's turned on in the neurons where it's needed. Its expression in the neurons, and it prevents the neurons from degenerating. And so when they follow patients, normally here's 23 patients they followed by their age, right, shortly after birth, and whether they were living or dying. And you can see every little step here is a kid dying from SMA because they can't breathe. And when they gave this virus a single dose, 91%, 20 out of 22 kids were alive and free of ventilation at 14 months here. And that resulted in, and it's long-term, we think. We're still following them out. It's been FDA approved for a few years now, but the kids are growing up, whereas before they had no chance. Really miraculous. So that was the first FDA approval of a systemic gene therapy. You're giving the virus into the vein, it's going to where it needs to go, and it's turning on the gene that's defective or missing and curing the defect. Pretty awesome, pretty awesome. Hemophilia is getting a little bit closer to my world. I'm trained in hematology and oncology. Hemophilia, we all know, is nasty bleeding disorder. Patients get lots of problems, not just from acute bleeds, but chronic joint destruction from chronic bleeding and so forth, head bleeds that can be fatal. Single dose of adeno-associated virus with the gene for hemophilia. You can see times 0 here and a measurement of their, in this case, factor 9. So hemophilia B, there's also factor 8, hemophilia A. But hemophilia B was the first one that was published. You can see the levels in each patient going up. You want normal people are between, usually 90 to 100%. But if you're above 5% of normal, that really stymies your bleeding. You don't need a ton to really convert from a severe patient with hemophilia to a very mild or no problems whatsoever. So you can see in all cases, you give this gene a single dose, and they all got really great levels of factor 9 expression, and they stopped having bleeding problems. Now, patients have been followed out for about 5 to 10 years now, and this is starting to fade. So there's a lot of question in the field is how long will, when will you need to do something different? This is not yet FDA approved because this is a disease where you already had a different treatment. We could replace the enzyme by giving it, but you have to give it every day to prevent bleeding, or multiple times a day to really get the factor levels up if they've had a trauma. So because there was another alternative available therapy, the bar for getting this FDA approved is higher than it was for the SMA or for the Leber's blind congenital blindness. There was no other treatments for those, so the FDA was much more lenient. But we're all hopeful that this will get approved soon, maybe even in weeks to months, because there's been a lot of research on this. So that's also very exciting. And then muscular dystrophy. My wife's a pediatric cardiologist. She's made her career to be a muscular dystrophy doc to address the heart, because it's all skeletal muscle and heart that goes bad in muscular dystrophy. And again, this is a situation where the kids seem normal at first. And as they start developing milestones, they start having more and more trouble and start getting muscle weakness because their muscles start breaking down because they're missing this gene or this protein called dystrophin. They have a mutation in it. And by their early teenage years, they've lost their ability to walk. And then soon thereafter, they lose the ability to breathe. And usually they don't make it past their 20s. So again, this was developed at our hospital, so I'm acutely aware of it and watching patients come through that are on the clinical trials. You can see here staining for the dystrophin gene in the muscle cells. And these are the first 4 patients that were in this trial that was published a couple of years ago. And you don't see any of the staining in the muscle cells. But after they got treatment, you see almost near normal staining on the bottom, which is very exciting. And these kids are walking and climbing stairs way beyond where they were expected to. Again, the FDA has a fairly high bar just because they want proof, because this is very expensive medication, but we're hopeful this will get approved. And there are some side effects to these treatments that have happened in some patients, so the field has to sort of deal with that and convince the FDA it's worth it. But again, where there was no hope. So I went to a meeting in about 2001 with my wife, went to this, they were hosting this group was called Parent Project Muscular Dystrophy, was hosting a big meeting, a banquet, end of the week meeting banquet. And I went with her to the dinner and met some of these kids and realized, there was a 7-year-old kid in front of me who was still walking, but realized he had no hope. I mean, at least the kids I took care of with cancer had some hope, but we might be able to cure them. But here's these kids, we know what's going to happen to them. They had no hope. And at the time, there was very little research going on. So it's been very exciting to watch that field, and a lot of people get in on a lot of drug development and gene therapy development. And we're very excited that this might become a nice treatment for them to improve their quality of life and their longevity. So there's a lot of different viruses being used in gene therapy for a lot of different indications. These are different kinds. Some are the kind that just put the gene in and aren't lytic, but some of them are lytic. There's a lot of activity going on. So I think we're just at the very beginning of an exciting field. We're using these non-lytic viruses to address this problem. So on this graph here, you see the number of cancer cells, logarithmic. So, 100, 10,000, million, so forth. As the cancer cells develop in your body, you finally get enough. By the time you're diagnosed, it's estimated to be 10 to the 9th or 10 to the 10th cells, and can cause worse and worse symptoms. And if you get up to 10 to the 12th, you don't make it. Obviously, with surgery, you can reduce that. And with chemotherapy, you start to reduce it a little bit. But with chemotherapy, you give it in pulses because the body can't take it continuously. And nor can we give drugs continuously very well. So every time you stop giving it, the cancer cells grow back, knock it down again, they grow back, knock it down again. So you got to keep, in order to cure someone, you got to treat them for several years until you get down to this zero. Because all these drugs have short half-lives, you got to give them repeatedly. And in some cases, we do hang continuous infusions. They have to walk around with a backpack, and that's very cumbersome with a tube hooked to their vein, puts them at risk for infection and so forth. So this all requires lots of hospital visits. Each dose only kills a fraction of the cells. And by pulsing it like that, you allow the cells to regrow and develop resistance. Like I say, we treat patients for a long period of time. So we came up with the idea that we could use gene therapy to produce a protein therapeutic with a single infusion for a long period of time, like is happening with the hemophilia, whereas that's replacing a defective gene. And all the examples I showed you so far are replacing defective genes in patient cells. Can we use this to actually not replace a gene, but to express a therapeutic? So instead of producing a protein therapeutic, some therapeutics are small molecule chemicals, some are proteins. So we can do this with proteins. So instead of making the protein in a manufacturing plant, shipping it off to the clinic, hooking it up to the patient IV and having it go into their body, where you have problems with stability, storage, handling, you need lots of infusions, you need pharmacy and nursing visits, or a hospital, clinic, or home care settings. We got the idea, why don't we take these small adeno-associated virus recombinants that have been shown to be safe for all these other diseases and engineer it to express a protein. Give it once, and basically you're turning the liver and other organs in the body into the manufacturing plant so that it continues to pump it out and give you steady state levels long-term, which might be good to fight cancer. So we produced a protein that's what we call bispecific protein. It's got pieces derived from other antibodies that bind on one end a tumor cell and on the other end a T cell of the immune system, which kills cells if it's contacted and linked to a tumor cell. So by that way, we can force the T cell to recognize a tumor cell and kill it. So we give it IV, ideally expressed in the liver and the muscle. And so we've just had this idea a few years ago, so we've been trying it out. We do get a single shot in mice. We get the levels to come up within 2 weeks, and then they're steady state for over a year. I don't show it all the way out here, but we've got data. And here's an example in our mice where we give the mice a tumor. On the back of the mice, you can see it light up here on this special camera. And if we don't do anything, These is what you see. The tumors grow and grow until we have to sacrifice the mice. But if we give that one treatment, this is what happens to them. They start to grow a little bit as the levels are climbing, and then those tumors all disappear. So we're hoping to put this out in the medical literature in the next couple of months as an idea. And we're spinning off a company because in order to have a drug get developed and taken all the way to the FDA and into the market, you really need to engage industry, and there's a lot of money. I think the average, cost of a drug developed these days is close to $2 billion and 15 years of time. So it's more than what us academics do. But we're spinning off this company, and whether it's going to work or not, we don't know. All these things can run into challenges. So that's when I was thinking of, the skepticism Rod might give me. Is that really going to work, Cripe? But I always made fun of Rod, so we're going to move forward with it and hope it works. So the other thing we're doing with viruses is trying to exploit the lytic type, the type that kills cells, as medical therapy, mainly for cancer, because cancer cells are the ones you want to kill. So, we want to shoot off the bad guys, in this case played by Izzy in our reenactment of classic movie. So this is what I'm talking about now. We call oncolytic viruses. Onco for cancer, lytic for kill. Oncolytic viruses. So that's the use of live viruses this time. The other viruses were sort of gutted or attenuated. We were just exploiting them to get a gene into a cell. This is live viruses to kill cells and induce an immune response against the tumor, which is a form of immunotherapy. And we can put genes in there and have them do different things. So that can also be a form of gene therapy, but it's basically a living drug. That means we put it in the patient and it starts doing things on its own. It starts spreading, starts killing cells, making more of it and spreading more throughout the tumor. And so in a dish, it might look something like this. You drop a cell you drop a virus into the middle of a whole culture of cells, and it just rolls through them like a tidal wave, killing all those cells. And I used to say this is my favorite movie, so I always like to show it twice. One virus in the middle, boom, spreads like a tidal wave, killing all the tumor cells. And that's what we want to see happen in patients. We certainly see that happen in mice, and I'll show you that in a minute. But this whole idea was actually started a long time ago by a guy named George Dock, who in 1860, well, he was born in 1860, died in 1951, and he was a professor and head of medicine and dean at WashU School of Medicine. But prior to that, he actually worked in Cleveland. And when he was in Cleveland in 1904, he reported this paper, The Influence of Complicating Diseases Upon Leukemia. And what that paper was, he reported a case of a patient of his, 42-year-old, that presented with leukemia. She had a big spleen. She had a low red count in her blood, a high white count, 367,000, a normal 10 to 12,000. They looked in her bone marrow, it was just packed with leukemia. And then she got the flu. And back then they didn't even know what a virus was, let alone that it was, that the flu was from a virus. That didn't really come until the early 1930s. But she got influenza. She got runny nose, cough, headache, fever. And she kept coming back to see him, and her leukemia seemed to disappear. Her white count was down to normal. She had a normal sized spleen. Her bone marrow exam showed no more leukemia cells. And that went on for another month or two. And then it started coming back, and she eventually died of leukemia. But he reported this case, and he said, it might seem that the process could be imitated, nature's experiment, essentially, and a symptomatic, if not a causal treatment, be discovered. So he is really the father of trying to infect tumors with viruses and get them to kill the tumor cells. And in the 20s, there were other investigators out in France who took herpes virus into epithelioma tumors grown in in rabbits, epithelioma surus, and they found that they could propagate the herpes virus, reculture it out of those tumors, and the tumors would shrink. And the virus still would cause keratitis, this is an eye, and encephalitis, this is a drawing of a brain. So it was a good virus. they could propagate it in these tumors. And so in the 1940s and 50s, actually, a number of viruses were used to treat patients. Hepatitis virus, West Nile virus. They picked sort of exotic viruses thinking that if patients weren't immune to them, they might do something. And they treated patients with different cancers. Not too many, 20s to 90. And they started getting some effects, whether they gave it intravenous or intramuscular or into the tumor. Some of the patients improved, some of the tumors shrank, some of them disappeared altogether. So it was pretty amazing, but these viruses cause infectious diseases because they're still bad viruses. And so patients got sick. And it was around that time that Sidney Farber started developing chemotherapy for cancer. And so this field kind of faded. Hey, the patients are getting sick. Even though it looks maybe promising, let's go after chemotherapy. And so this kind of fell out of favor until the 1990s when Bob Martuzza up at Mass General started playing around with herpes virus and was able to engineer it to not cause these sores. So the versions that he made had deletions in them. They call it attenuated virus, kind of like a live virus vaccine, wouldn't cause these sores, wouldn't get patients sick, but could still infect and kill tumor cells. So this is one of the viruses that it's actually a medium-sized virus. It's complicated, lots of proteins inside an envelope, lots of what we call glycoproteins on the outside that interact with cells in different ways. The genome's pretty long, 150,000 base pairs in length. It's got different structures, but he made certain mutations of the genes that we could do by the 1990s to make them safe. And we took one of these viruses and we were growing tumors in mice. And this is a pretty nasty tumor in a mouse. And we gave it a virus and you can see the tumor's just, it's giving the tumor an ulcer and shrinking it. And we could get cures in these mice. And this is an example of the tumor growth. If you didn't do anything, or if we inactivated the virus, made it not a live virus, we exposed it to ultraviolet radiation, tumors would grow. But if we gave a single dose of the virus, we could get pretty large tumors to shrink and disappear. 7 out of 7 complete responses in this example of a muscle tumor that kids get, rhabdomyosarcoma. So that was very exciting. We went to clinical trials. Others went to clinical trials. Things don't always work as well in the patients as in the mice, but in one case, there was a study that was done with a herpes virus they called TVEC in adults with melanoma. They injected them into the lesions on the skin and compared it to a protein injection, GM-CSF protein, which was another treatment at the time. And they showed what we call waterfall plots. The waterfall plot shows what happens to the tumor growth after that injection, after that treatment. Does it grow or does it shrink? And they plot that with maximum growth in a patient or maximum shrinkage. You can see why they call it a waterfall plot. Each line here is a different patient's tumor and different stages and so forth. And 26% of the patients responded. That means they went 50% shrinkage or more. And you can see all these patients down here went 100% shrinkage compared to only 6% who got that protein. And if they looked at just each lesion, so that's overall, how the patients do overall. If they looked at each lesion, it's every line, there's so many lines here, it looks like just solid areas. And you can see the numbers here. This is a graph of 2,116 injected lesions. And 47% of them, almost half of them, disappeared, 100% shrinkage. 65% shrank more than half. You can see this. So these is all the ones that disappeared altogether. What was remarkable about this study was shown on the next two slides, and that is even the uninjected lesions shrank. Not to the same degree, but you can see 22% of the lesions that were around there that they didn't touch disappeared. 33% shrank at least 50%. And furthermore, in the viscera, that means liver, kidney, lung, tumors that had spread elsewhere, they're injected in the skin and they're getting some shrinkage and even some total disappearance. 9% of the visceral lesions they measured disappeared completely. So that speaks to what we call an abscopal effect. It means you're inducing an effect in something that you're not touching, and it's been shown now it's due to an immune response. By injecting the melanoma with this virus, you cause an inflammation, the immune system responds to it, and the immune system also goes and attacks tumors in other locations. So that's pretty remarkable. It wasn't curative in everybody, but here was the protein survival curves. Every one of these is a patient death, and patients who got the virus did much better than other people. And if they did well, if they responded in the first year, 12 months, they were going to be basically cured, as opposed to those who didn't respond before all the way to 12 months, they kept having problems with their disease. So I, at the time, was on the FDA advisory committee to reevaluate all these data. And because this was, again, although it was developed by an academic, it was licensed to a company to develop and bring to the FDA, Amgen. This is when I was walking in the room. So there weren't a lot of people there yet. This is the room at the FDA outside of DC. This is the tables up here where the committee sits. This was in 2015. I was on the Cellular Tissue and Gene Therapy Advisory Committee. The Oncologic Drugs Advisory Committee joined in as well. Had a couple-day meeting on this. And here's a view from my seat. I'd kind of held my iPhone just above the table, trying to be inconspicuous, took a little camera shot, and you can see I wasn't taking a lot of notes, but believe me, I was paying attention. And it was a very exciting time to see these data. And this is a lot of the company folks over here. And I was just amazed at how well prepared these companies are. We came up and peppered them with question after question after question after their presentation. And within a second or two of us asking the question, and sometimes even before we finished asking the question, they had a new slide up, randomly picked from, not randomly, but selectively picked from their slide decks that had the answer to the question. It was kind of an amazing sight. 22 of us voted in favor. One person voted opposed, and the FDA approved the drug as a treatment for melanoma soon thereafter. So that was pretty cool, but that was 7 years ago. And no other oncolytic virus has been FDA approved in the United States for any cancers yet. But there's a lot in clinical trials and a lot still in the laboratory. And things don't always work as well as they do in the mouse. You have to go back and try things in the lab. And so that's the stage we're at right now. We're tinkering in the lab, trying to figure out how to make them better, engineer them before we go back to the clinic to try to get them approved for some of the cancers in kids, which is my goal. But I think there's still a lot of hope in this field because there's a lot of activity going on. So the Greenbrier, myself, Marianne Flood, Doran Burrell, and Alan Wayne here eating. And I've just pulled this picture out because, I mean, just check this dude out here. I mean, is that not the coolest cat that's ever gone to science camp? And I call out Alan specifically because he's one of the other science campers that I know of that went into the field that I went into, pediatric hematology, oncology. And Alan and I have had the pleasure of getting together frequently and at meetings and staying in touch and advising each other. And before he moved out to LA to take the job there, he called me up to see how being a division chief was and whether, you know, bounce some ideas off me. And it's been a real pleasure to stay in touch with him. And he still is looking almost as cool. So in summary, you all know NYSC was inspiring, impactful, and fun. And I thank the Alumni Association for the opportunity to reconnect with you guys and gals. And just to summarize my science, viruses can be used for gene therapy to treat diseases, and they can be used as an oncolytic therapy to treat cancer. So my conclusion is that due to human ingenuity, viruses have a good side. So in closing, I pulled out my yearbook from 1978, and we had elected Rob Harvey as our alumni president, since this is Alumni Association presentation. And I underlined the end of his letter that he posted in there. I ask you, I challenge you, I dare you to use your potential for good and be an inspiration to others. So that's what I try to do every day. Still working on it, but and I encourage everyone listening and watching this in the future to take Rob's words to heart and continue to carry on the spirit of camp. And just to get a little philosophical here at the end, in my mind, the secret to success is actually failure. There are so many things along the way that haven't worked. And I touched a little bit on them. I mean, I showed you mostly successes, but behind each of those successes, there's a lot of stuff that just doesn't work. I always tell my trainees, if 10 to 20% of what you do in the research laboratory works, you're doing great. I mean, on any given day, you drop something, you screw something up, or something just doesn't work the way you thought. You got to go back to the drawing board. Or in life, you make mistakes. But you have to, it's Miss Frizzle from the cartoon my kids used to watch, get messy, make mistakes. That's what you have to do to be successful. But in order to, live through the failures, and I do think you need a couple of attributes. That's resiliency and humility. And I've tried to cultivate those attributes in order to get through all the failures to get through some successes. So for any young people out there, this is my philosophical advice to you. And I got to thank my family for helping me be resilient and helping me get through the failures. This is a picture of my oldest son, Kevin, from his wedding to Allie right before the pandemic. Here's Linda and I, and my son, Jeff, and his now wife, Jordan, and my daughter, Natalie, and her husband, Sam. Both of these couples weren't married at the time, but both of them got married during the pandemic. So we had 3 weddings in 2 years, and it was a lot of work, but we pulled it off. And now my grandson here, Hayes, we've been watching the Bengals in the Super Bowl recently, and that's what keeps me going is these guys. So thank you to my family, and thank you to the NYSC family for providing such fun and inspiration back then and through all these years. So that's it. I appreciate you guys listening. I'm happy to have a chat, take questions. Appreciate your attention.
Hey, thank you very much. That was awesome. Is there stuff here that's too proprietary to share with the national camp this summer in a virtual lecture?
No, everything there is public.
Let's talk. I just sent you a LinkedIn invite. I'm the director of the camp. It might be really cool to have this idea at camp this summer. I really enjoyed. Thanks. I'm Brian Kinghorn. I'm the director of the camp. Sorry I was a little late joining, but fascinating stuff.
No, I appreciate the invite. Yeah, I've always wondered, I've always thought, someday I ought to go back to camp and give a talk or something, but, life gets in the way and every summer comes and goes. But I think that'd be a lot of fun. I'm not too far away.
Well, we're virtual this year, unfortunately, so it doesn't matter how far you are. Yeah, let's talk. I don't want to monopolize the opportunity for questions here, but this was really, really cool.
Thank you. Happy to
I
check.
think we have to not forget that there's a reunion next year, even if we hope camp will be in person, and I really hope that too. But even if they don't, we'll have a 60th reunion of the beginning of the science camp. And we'd like to see Tim pass the word around to all you, Alan, and all of your friends, see if we can get as many as possible to make that trek. And Ray and I will work on getting transportation from the nearest airport to Hilltop in West Virginia. And it was a wonderful talk. I learned a lot. I'm not in medicine anymore, and I retired at age 75. But it was a great talk, and I learned a lot. Thank you.
Thanks, Rick. I appreciate it. It's always great to see you.
I remember seeing Tim at one of the times that he was on staff and I came back to lecture, and he said he remembered the talk that I gave at science camp. And he said, if that dumb guy can become a doctor, I can do better. And he did.
I think you're putting words in my mouth.
but it is a great brotherhood of, I mean, I see Woody here and how many years we go back, and Bill Hilton's watching. It's just, you can't even describe it to other people how, and you hit the nail on the head. We were at a very, not a vulnerable age. I don't want to say the word vulnerable, inspirational age. They're learning to be humble in the face of all the other people who are so smart, and then act silly and yet be inspired to just keep doing your best day after day. And you're a great example of that.
Thank you. That's all you can do, you know, do your best. Appreciate that.
Hey, this is Lisa. I was a camper way back in 1998, and I have a question for you. I'm sort of on the other end of the development of what you do, and I work for Big Pharma, helping to develop drugs. And so I was wondering, with viruses, what do you think is the biggest challenge to actually being able to have this work in a clinical setting and actually getting to a drug that will work and pass muster?
Yeah, so for the gene therapy ones, again, there's 2 examples now that are FDA approved. And I think the challenge in the field right now is the balance of, well, 3 things really. Obviously safety and efficacy, but also cost. These are not cheap to make. And the cure that I talked about for SMA, spinal muscular atrophy, it's a one-time dose, but the charge that'll cost you is $2.6 million. But the argument is you save the life, it's a one-time treatment. But you know, that's not tenable, especially to spread around the world. But I do think the engineering, the production of these is going to reduce the cost. As these have been used more and more, there are side effects, just like we found out there's some side effects on rare occasions with COVID vaccine. You can induce inflammation in patients. If you have an underlying liver problem, you can get liver damage at really, really high doses. Some of these things cause a systemic complement activation. So, and there have been a few deaths on the trial. So, but as we learn more and more, then we learn ways to circumvent that. So I think it's just a matter of figuring out navigating those waters of making sure you can maintain good efficacy, but avoid toxicity and get reduced costs. So, you know, it's really, the gene therapy and the oncolytic virus fields are really still in their infancy, but we've had some successes to date. And I think that gives me hope that we'll be able to figure these things out. I've been constantly amazed by how ingenious people are. I go to these national meetings, and just like Rick said, other campers, but all the other scientists, you know, people are brilliant. They come up with crazy ideas that sometimes work. So it's, I have hope.
Hey, Tim, it's David Harden from your 1978 camper year. Hey, great to see you. And yeah, we always knew Tim was headed straight for medical school. There was no question about that. He was just one of those guys that you just knew. He was one of those guys, as Rick was saying, you go to science camp and first day it's like, oh, all these people are smarter than me. And then the second day it's like, oh, all these people are as goofy as I am. And by the third day, yeah, you're thick as thieves and everything's great. And so Tim was one of those people that I remember meeting early on and saying, yeah, this is a guy who's going to do things. And glad to see you and keep up with your career. So the question I had was, there was a lot of talk in the wake of the development of these mRNA vaccines for COVID that there might be significant uses for this kind of technology in oncology. And I was wondering if you had any thoughts on that. I know it's a little perpendicular to what you're doing, but I was just interested to know if that chatter was just idle speculation or whether there was anything to it.
That chatter is absolutely spot on. And I think that technology is going to be fantastic. I'll tell you a story. I was reviewing a grant before the pandemic submitted by a group that was using RNA nanoparticles to try to create cancer vaccines. And one of the other reviewers on the panel kept making the argument, but look, he kept saying, because I was saying, well, this looks promising. His data in the mice look pretty good. Let's, support him to do the clinical trial. And the other person who's senior to me, was kept saying, well, nobody's ever shown RNA vaccines can do anything. So after Moderna and Pfizer came out with their vaccines and they're saving the planet from the pandemic, sort of too slowly, but you know, game changer. The person sent their grant application back in the same committee, and now we're like, yeah, this is good stuff. Let's get it through. So it was all in the timing for him because they proved that this technology can work. Now there's always going to be nuances and lots of things to figure out, and how best to use them, how to give them to the patient, but the basic idea is the same. Can you activate the immune system against not a virus target, but a cancer target? And can you get then the immune system, leverage it, parlay it into killing the tumor instead of not? And that's tricky because you got to have the right target to go after on the tumor that doesn't kill normal cells. And tumors are derived from normal cells. So a lot of what they have on them or in them is normal, but there are differences and some things you can exploit. And it may not work for all cancers, but I'm confident that technology, and I think Moderna, at least, if not Pfizer, already has some clinical trials of their mRNA nanoparticles in cancer. So I do think that is the upside. Again, if you want to say good thing of viruses, the upside of the pandemic is there's a lot of technologies that were proven to be useful that I think we can apply in other settings.
Also goes to show in your example that you can be ahead of your time a little bit.
Absolutely. You got to, sometimes the technology or the field has to catch up with your ideas.
Yeah. Anyway, I was saying to Rick, I will be at this 60th anniversary reunion in 2023, and I challenge all my 1978 camper colleagues to do the same.
Good challenge. I see Rob Harvey on too. Maybe he'll
take Hi, Rob.
Good to see you guys. Tim, I just wanted to say it's wonderful to see you doing something impactful. And I don't remember that line, but I'm not surprised that I wrote it when I was young and idealistic. Then I became a lawyer. I learned the word Dave Harden at our talent show, as I recall. Tim came out stripped half naked during that talent show. It's one of the MCs, it's my memory, but Dave did a skit called Terpsichore, and I have to keep going and look up that word because I never remember exactly what it means, and it never means what I thought I remembered. Tim, quick question. Setting aside all the mixed messaging and terrible messaging we got about the vaccines and COVID, causing all of us, many of us, to lose confidence in the national government's statements about what to do and what to wear and how many masks to put on. President Biden has announced this new campaign against cancer. Is there a Project Warp Speed that you can get involved in or that's on the cusp that will work for cancer?
Yeah, absolutely. I think, Biden's original moonshot program, as they call the cancer moonshot, that he launched when he was the vice president is there was a big chunk of money put into it at the time, and we were fortunate enough, they carved out a pediatric component, and we were fortunate enough to get a grant out of that we're still using. And we're hoping that with this new boosting of that moonshot to use some metaphors that go together, we hope that we'll be able to continue to leverage that money and develop some of these technologies and develop, more potent and safer cancers. It's a long slog. These things take time, way too long. When I'm giving talks to family groups or private foundations that have formed from parents who have lost kids, it's never fast enough by any means. But there's just so many unknowns and challenges, and you have to work through it. But you have to be perseverant and, keep going and not let you got to be resilient, not let the failures get you down. So maybe 2 steps forwards, 3 steps back, but that might be followed with 4 steps forward. So ultimately, I do have a lot of hope.
I have a follow-up question on that. You were talking about creating your own drug company to work on R&D. What are the pros and cons or the benefits of starting from scratch versus kind of inserting this idea into an existing company, where you wouldn't have to start at the ground level. And again, I'm speaking from a lack of experience here. This is just an honest question. What are the pros of this decision to start something brand new as opposed to working it into something that's already existing and might have some of the structure in place that this might happen faster?
Yeah, it's a great question. It's all about risk. So the big pharma usually isn't willing to take extreme risk and invest in things very, very early that someone else is developing. They have their own R&D, their own priorities, but they're not willing in most cases to put a lot of resources into things that are very, very early on because they're unproven and the majority of things fail. And so they tend to want to take something on, and obviously there's exceptions, and I don't want to misspeak, but they tend not to want to take things on. Whereas venture capitalists who are willing to invest in very early stage, they're willing to take those risks. And the reason is because when they invest at early stage, they get a bigger percentage of any future royalties or revenues. So they also know that these are risky, but that there's different venture capitalists that's the business they're in. They say, well, we're going to pick 10 things to invest in each year, and we know 9 of them aren't going to work, but hopefully the one that works is going to more than make up for the others that we lost out on. So the advantage is that you can get chunks of money from people that are willing to take those risks, whereas something this early, it's just hard to get the attention of big pharma because they got so many things going on and so many priorities.
Does the university, the Ohio State University, also provide support for setting up these venture type efforts?
Yeah, most of the time universities are, yes, most of the time universities are supportive because they'll get in on it a little bit and maybe get a return on their investment. So in my case, it's actually Nationwide Children's Hospital, even though we're academically affiliated with Ohio State, it's a separate legal and financial entity. And so they've given me some money to continue to develop it. And they're also helped me find some business partners that I would partner with, because that's not what I do. Then it's their job to go raise money and sort of work with pharma to, once things get a later stage, hand it off to big pharma to keep developing. And so yeah, they get a little cut too, and so, but it's win-win for them. And for me, I spent most of my career developing things and publishing papers, but not taking them to the patients. But when we had this cool thing, I'm like, this has to get to the patients. And what's the quickest way to get it to patients is to attract bigger money and get it out of the academic setting.
Is there a popular novel for us non-medical, non-biological folks to read? I'm thinking of something like, what is it, Code Breakers, that talks about Doudna and Charpentier's Nobel Prize work on RNA.
Yeah, that's with CRISPR.
Yeah,
the gene editing technology that also is sort of groundbreaking. I didn't talk about that. We're doing a little bit of it, not a lot, but there's some really cool stuff going on with that as well. And I think that's a big part of the future, gene editing, just actually going in and changing base pairs in the genome. And right now they're doing it in cells that you can pull out, like bone marrow cells, CRISPR, edit them in the dish, and then give them back to the patient. We have a trial we're about to join here where we do that with sickle cell disease. So very exciting stuff.
Very cool. I may have asked you this before. Did you ever meet Dr. Stasny in the stat department? Her sister was on staff, and
No.
she just retired, I think, from the statistics department there at Ohio State.
It's small world, but no, I don't recall that name. You've wasted another good hour, as they used to say on NPR. Shall we call it, Ray? Oh, you're on mute, my friend.
Still on mute.
Yeah. That's the the bane of Zoom, right? Figuring all the buttons to press.
I stuck a quick question in chat. Is it possible to get the slide deck?
Yeah, sure. I'm happy to. It's a pretty big file. I might have to send it as a PDF. Should I email it to you, Ray, and you can, or do you want to? If you guys want to just email me or Ray, and I can happy to send it out to you.
Yes.
Again, well, I'm not very good at reading lips, Ray.
Since I'm probably just, if Tim can send them to me, I can distribute them to anybody that wants them that way. He's a lot busier than I am, so I don't want to take up any more of his valuable time. We have to. So if anybody wants stuff, just let me know. And then of course, I think I stated at the beginning of this, this is recorded and it'll be posted on our Laura Dill NYSCAA YouTube channel here in the next day or two. So, but anyway, if you want to just go ahead and send them to me, and then anybody that wants them, I'll be happy to forward them on to you. So,
sounds good. Thanks again for the invite. It was fun to put this together.
Well, thank you, and thanks for all the wonderful trips down memory lane with some of those slides too.
You bet. Thanks for dialing in. Good to see you all.
All right.
Thanks on behalf of the cancer survivor community.
Sounds good.
Yeah. Now I got to bug my nephew to see if he ever had you as a professor at OSU. He's got a different last name than mine and it's generic. So I'll just email him rather than asking you.
Sounds good.
Okay, great. Take care.
Take care.
Bye-bye.